All right,
Got it.
great. Leona Clark-Harvey, Maria Bonneville,
Present.
Mark Fisher Coldry,
Here.
Fred Fisher, Judy Gatson,
Here.
Larry Goldstein,
Here.
David Higgins,
Here.
Vito Imbasciani,
Here.
Steve Julesgaard,
Present.
Pat Levitt,
Here.
Lauren Miller Rogan,
Here.
Marvin Southard, Keith Yamamoto. Okay, we do have a quorum and we can proceed, Larry.
Great. Thank you very much. So today's meeting will primarily be focused on asking questions about the CIRM portfolio and the degree to which we want to do planning of that portfolio. So the first hour or so will be a deeper dive into CIRM's past funding behavior that's been put together by the CIRM team. Those of you who've had a chance to glance at it know that it's a pretty impressive dive and provides a lot of interesting information. And then the second hour, I guess what I'd like to talk about is if there's $1.5 billion set aside, just as an argument for the amount, what fraction of that do we want to program by action of the board and the CIRM team together versus continuing to allow the grants working group to make decisions based on priority as applications come in for broad RFAs? And this is a question any funding agency faces. How much centralized planning versus how much distributed review and priority setting by grant review? So two sorts of pithy issues to get us through the day. And so let's go to the analysis of the CIRM portfolio. Who's going to present that?
I will present Dr. Goldstein on behalf of the CIRM team, represented by Dr. Chrissy's team and my team.
Thank you.
So, Marianne, if you would like to put the slides up, please. Thank you. Let me just. So go to the next slide. As Dr. Goldstein was saying, this presentation is a deep analysis and gap analysis of our portfolio. And this is always in the context of CIRM's, achieving CIRM's mission to accelerate world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and the world. Next slide, Marianne. So these are the questions that many of you are familiar with that we are going to be answering. So I'm going to just go through them very quickly here, and then before every analysis, we will place the question. So we are going to go over what is the percentage of DISC, TRAN, and CLEAN awards that are within the Neuro portfolio from the 2007 to 2023. So that could include Prop 14 and 71. But we also did a sub-analysis that shows just Prop 71 from one side and just Prop 14, so that we can see whether there are differences since we started funding. The percent of Neuro DISC awards that have progressed to TRAN or CLEAN, what's the progression that has that we get from our early funding? What is the percentage of DISC awards that are Neuro relative to disease burden? And we did the same for TRAN and CLEAN. And then we are going to look at the distribution of Neuro awards relative to the geography of our academic and medical research institutions. So for the first question, next slide, Marianne, thanks. The percentage of these TRAN and CLEAN awards that are Neuro. Next. This is for Prop 14 only, which corresponds to the last nearly 2 years. Next slide. What we can see in this slide is that 37% of the discovery awards for Prop 14 have corresponded to Neuro awards, and that corresponds to 34 awards, as you can see on the little table on the right, out of a total of 91. The second bar corresponds to translational. The 27 awards corresponding to 9 total awards out of the 33 correspond to Neuro. And then for clinical is 27% as well, which is 10 awards out of 37. If you want to go to the next slide, this is for Prop 14. So we are going to see whether this is the CERM. One second. This is supposed to be, oh, that's in funds, sorry. And this, the last slide was the number of awards. And this is the percentage of funds. So the top of the graph says percentage of total awards is actually the percentage of funds that we have spent in this TRAN and CLEAN in Prop 14. So for this is a 37% of the funding, so $50 million that we have spent so far in Neuro. TRAN is 26% of funding, $39 million, and CLEAN 28%, which is $86 million. And now we're going to see the Prop 71 analysis. Next slide. Next. This actually is the total, whether, and what we want to see here is whether there's been much of a difference between what we've been doing since we restarted Prop 14 and what we had done so far. And the overall conclusion, if you compare this slide to the one that was the number of awards, is that there is not much difference in the way we are funding now to what we were funding before. So we are doing about the same amount of awards. So 28% of awards, or 196 total awards, were corresponding to discovery. For translational, 33 of the awards, which is actually 34%. And for CLEAN was 22%, 30 awards that corresponded to Neuro. It's a little bit more TRAN in this case than in what we are doing now, but I will leave that. I think that's considered more or less the same. The next one is going to be how much money have we spent, how much funding have we spent in Prop 71 and Prop 14 for Neuro. Next slide. In terms of funding, we have spent $362 million in discovery in Neuro. That's about 31% of the funding. 35% of funding, $204 million for TRAN, and 29% for CLEAN, or $367 million that we spent. So as you can see, we spent about the same amount for TRAN and CLEAN in Neuro in the past. Any questions so far? I don't know if we want to take questions now, perhaps at the end, Dr. Goldstein.
We should take questions at the end. Let me just interject one point. If you ask what fraction of 5.5 billion is the 1.5 billion set aside, the answer is 27%. So the numbers you're seeing that are greater than 27%, which is most of them, that actually, in a sense, reflects having completely fulfilled the mission so far just by doing nothing other than broad RFAs and selecting the best proposals based on that. Sorry, Rosa, go ahead.
Thank you. No, that was a great comment. Thank you, Dr. Goldstein. So now, what we're going to look at is the progression of the funding from discovery. What is that we basically de-risked to go into TRAN and CLEAN progression. So next slide, we can see that there are 28, Marianne? There are 28 projects that progress in total, in the total portfolio of CERM from some stage to TRAN or CLEAN in total. Of those 28, 15 of them went from discovery to tran and clean. And you can see them as the discovery would be the green boxes. And of those, there are 15 that go either from a discovery idea, discovery candidate to tran, or to clean, or to tran and clean. So of those, we kind of de-risked internally 15 of them. And that corresponds to 2% of the total discovery awards, i.e. 689 awards. That's the total of discovery awards. So 2% is the 15. And the color coding is the indication. So the highest number has been that we have seen in terms of progression has been neurodegeneration, Parkinson's, ALS, Huntington's, and then we have a few in eye, like as indicated by the retinitis pigmentosa and a macular degeneration award, but that's from tran to clean, it's not from discovery. And then we have some in spinal cord injury and one in traumatic brain injury. And then we have 2 in epilepsy. And then we have actually only 1 in epilepsy that went from early discovery idea to candidate to clean 1 and clean 2 directly. It kind of bypassed the tran. And that's actually a collaboration with a company between academia and then companies, Neurona Therapeutics. And then we have one that's in Canavan disease, and that went from discovery candidate to translational. So the next slide is going to show us the percentage of DISC awards that are neuro relative to the disease burden. So how much, and then we did the same for the percentage of tran and clean awards relative to disease burden in the population. So we are going to see how much have we funded. The disease burden we calculated is represented here by the DALI, which is the Disability Adjusted Life Year, which is a representation of the loss of the equivalent of 1 year of full health. And these numbers are up to date, and they include global health and WHO numbers. So the CIRM funding for discovery here, we had the highest funding was in Parkinson's disease with $51 million, going into neurotrauma, another ALS, eye diseases, Alzheimer's, and then going down. What happened? Is it me or is?
No, it's a presentation.
Oh, okay, got it. Yeah, because I'm actually in a non-ideal setting, so I wasn't sure. Thank you, Mr. Jorgens. So that's what we were looking at. And then, as you can see, if you map this to the DALI, we can see that for Parkinson's disease, the disease burden is relatively lower than, say, stroke, as I said, as represented by the loss of equivalent of 1 year of full health. But then we can see that we have a very little funding for some diseases that have a very strong burden, as you can see, for example, at the bottom, with neuropsychiatric diseases and their burden. Next slide is going to show the disease burden for tran and clean awards. So for tran, we can see, next slide, Marianne. For tran, you can see that the highest amount of funding was for brain cancer and eye disorders, followed by Alzheimer's disease and stroke and other neurodevelopmental disorders. And then we have trauma and Huntington's disease, epilepsy. But again, we have relatively good amount of funding in diseases that have a lower DALI burden. But then for those that have highest, we like, for example, autism spectrum disorders, we have nothing. It's not as a higher burden, but then again, neuropsychiatric looks very low. There is none. The next one, and that's also probably because of the state of the science at the time as well, right? And for translational and clinical neuropsychiatric, there might not be much that we can do. However, we might be able to do more for discovery, as we've already discussed. Next slide is for clean. Next, okay. And here again, we have a bit of a switch from discovery, in which brain cancer and eye disorders, as well as some neurodegenerative disorders like ALS and neurotrauma and stroke are like the highest funding. And again, we can see that there is a low level of funding. Surprisingly, for Alzheimer's disease, there's been discovery and translational, but there's nothing here in the clinical. But obviously, you know, there is in terms of cell therapies or gene therapies, we still don't have anything for Alzheimer's disease. So I'm not surprised here. It's more small molecules. Next slide, and immunotherapies. Now we're going to see the distribution of neuro awards relative to the geography of academic and medical research institutions. So for discovery, we can see that the highest amount of funding, the highest number of awards is actually in the South, in the San Diego area, followed by the San Francisco Bay Area, and then Los Angeles and Orange County. The Shasta, Sacramento area has about 4, followed by Central Coast and the Inland Empire, and outside of California, there is 1 award. The next slide shows for the TRAN awards. The Bay Area is the one that has the highest number of awards. San Diego, followed by San Diego, Los Angeles, and Orange County, and then again Shasta, Sacramento, with the Central Coast and the Inland Empire that don't have as many awards. And then the next slide is the clinical number of awards. The Los Angeles area is the one that has the highest clinical neuro award number in California, followed by the Bay Area and Orange County, the Shasta, Sacramento, and then outside California, there are 2 awards, and San Diego area has 2 awards. And then I think now we are going to go into the questions, but I think Dr. Goldstein already, Marianne, you want to move the slides to the next? Right. So the 3 questions, I think I'm going to leave it to you, Dr. Goldstein.
Yeah, let's hold off on these. Yeah, thank you, Rosa. Let's hold off on these for the time being. So first of all, thank you, Rosa, for the great presentation, and thank you to all the different members of the CIRM staff who pulled these numbers together. This was a good, solid analysis of the data that we currently have, and that actually pose and some interesting questions. So first off, questions about the data themselves from the group, please. Mark.
Hey, yeah, terrific data, well summarized. Data all generates additional questions. The neuroprogression slide looks really interesting to me in the context of, I'd be really intrigued about whether some of those things that are on that progression slide has further opportunities for future progression, or have things come to a halt. And if they have come to a halt, is it, you know, because the scientific advancement is not possible, or is it related to funding? So I don't know if there's, you know, a way without expending an inordinate amount of time, you know, to understand that, because obviously within our context, we're trying to move things forward on a continuous basis from start to finish. It'd be really interesting to see if there's still opportunities for some of those on the progression slide to continue forward.
Yeah, I would just add, Mark, it's a good point because we don't have data compiled yet on projects that went, say, from DISC into private industry, or
Right.
that traveled with, people who left the lab to go somewhere else. We have a drug candidate that emerged that went with an ex-postdoc of mine to the Netherlands, which is where the trials are going to be. So it's not that things don't progress medically or scientifically necessarily. Sometimes we just don't know where they went. Steve.
Yes, so I'm struck on slide 13, which shows the neuroprogression by disease area, that what we listed in the second column are all institutions, and I don't see any industry involvement in any of these particular areas. And yet we've been funding industry in a number of areas. So isn't that industry is just not asking for money from CIRM for the neurological area, or what do you think the issue is that we're only seeing institutional involvement? There, that's the slide. Second column over, institution.
Yes, so I can, there is, one of the questions that was floated was how much of this portfolio has been the risk that has gone from, say, discovery into translational clinical, but outside, like, say, industry. And I don't know if our colleague from business development, Dr. Shyam Patel, is on the line, and whether we have data on this, but I would say, is he there? I don't know if he's there.
Not.
No, he's not. Okay, we can actually get back to you on this. My first answer, my suspicion is that there is very little, because we would have recorded that. So I think the areas, the kind of funding that we have the risk is mostly has been going to academic institutions, as is shown in this slide. And we will get you the numbers if there are any outside the risk team.
All right, just a follow-on question, Rosa, and maybe actually for somebody else in the room. So I know if I go back to what I call the old days, let's say I look back at 2016 and the grants we were making in those days, the grants that were being made were actually in excess of the grant limit. So for example, we might have a translational limit back in those days of $5 million, and yet a grant was made in the neighborhood of $7.6 million. And while I don't recall for sure, I assume that the difference was the overhead part associated with an academic institution that drove the amount of money up, in this case from, say, 5 to 7.5 or 7.6 million. Is that the way we're still providing support? Is we have a standard grant amount, but then sort of in the background, there's the administrative support or overhead component, which really isn't in the numbers that we approve. Does anybody know that?
Public, would you like?
Okay.
No.
Go ahead.
Okay, yes, the train grants and CLIN grants include the overhead, just like I've been with them since 2016. All that, what you just described, Mr. Joscard, is included, so nothing's changed.
Well, if you, for example, go back to, if I look at what was done, what date was this? On March 16th of 2016, and you look at the awards that were being made, and just look at that in particular. Let me bring up the summaries for a moment. The one that got the highest score, which was 95, the budget amount that was approved was $7.377 million. There was another one that scored 85 that got $7.659 million. And yet the slides for that presentation showed the maximum amount for this Tran award of being in those days $5 million. So I'm just trying to make sense of what we did back then and whether we're doing the same thing today.
Whatever. We have Jen Lewis here, who's our head of grants management. I'm going to repeat the question.
I heard it.
Okay, good.
So Steve, I think you're right. So I think the difference is in our translation, you're correct that in our translation program, we have a total cap on our direct cost amount. And then on top of that, awardees can ask for any eligible overhead. So that's why you might see a variance between the numbers. In CLIN, that is the only program where we have a total award cap. So overhead is included in that total award cap.
Got it. So these days, when we approve TRAN awards, we don't see that overhead amount shown in the awards we approved. We're just seeing the base amount.
No, you'll see the total award amount will include both the direct and the overhead amount when you're approving those grants.
Huh, okay. I don't recall seeing that. Anyway, one of the points I wanted to make is that when I look back at what was happening way back in 2016, and I want to remind people that costs were a lot lower in 2016 than they are now in 2023 in all sorts of areas, but that includes the area of research and clinical development. Some of the most, the grants with the greatest amounts of money were actually in the neurological area. So there was a bigger ask for grants that involve diseases involved with that issue. And it brings me back around to a discussion that we had, earlier when I forget the researcher from Stanford, but we asked, why he hadn't applied for research grants from CIRM, and he said, well, it's just not worth taking the time to do it because the amount of money that's involved is just not adequate for the kind of work that I need to do. And I've been trying to figure out, kind of where we've been and where and what we've been doing in the meantime. And I haven't been able to do as much work as I'd like to kind of look back over time, but I'm not sure we have changed the size of our grant amounts from maybe, let's say, 2016 to today by any appreciable amount, if we've even increased them at all. And I think, and that this comment may apply more generally than just to the neurological area, but I think it's one of those things that we need to have a look at as to whether or not, we're kind of keeping up with the times, so to speak. I do know on the industry side, from personal experience in dealing with people in industry, the costs certainly have increased for both materials and supplies, for labor, for clinical research organizations. There have been some appreciable cost increases in this day and age. So anyway, it was just what I had an interest in and question about is whether, and it goes all back to the question of whether or not we're providing adequate funding here in the neuro area for any of these diseases. It's something I think we need to take a good look at. Thank you.
Thank you, Steve. That's actually quite
So-
persistent. Let me just make a comment, and then yes, Roselle, I'll pass it on to you. One thing to be thinking about throughout this conversation is, do we want our allocations of funding to be matched to disease burden, disease frequency, disease cost, disease disruption of people's lives? This is, again, a standard set of questions that funding agencies cope with. Rosa.
No, I was just going to comment and clarify that we've been hearing this comment and we are aware and we are working on this. And at least for the new concept that is coming in September, we are taking this into account, at least for the neuro strategy that has to do with discovery. So that the model that we would be and the structure that we would be presenting would be reflecting this. Just wanted to make that comment, that we are hearing it loud and clear.
Great, thank you. Mark.
Yeah, my question has to do with CIRM had to choke off a bunch of funding as funding was running down, and a lot of researchers were discouraged from filing applications. And when I went back to revisit people upon joining CIRM, they had interestingly enough, even though they've been prior grant awardees, were way behind the times on where CERN was at, what the status was, what was going on with the addition of, cell therapy, the size of the grants and the sequencing. And it's like, oh, you guys are back open for business. They had no acknowledgement. And we're talking, surprisingly, some individuals at UCSF and Stanford and a lot of other places. And the reason for that intro, and it gets back to the last question, is I suspect there's a vast amount of misinformation running around about dollar amounts and how hard it is to file an application, because for a while researchers were discouraged from filing certain types of applications because of the dwindling funds, and therefore were directed not to do that in a way. That's not exactly the right terminology, but I believe they were discouraged. So the corollary question I have, again, getting back to disease progression, is the phenomenon: did we choke off any grants that people had applied for, but we were out of funding, or were dwindling down on funding, or funding got redirected to other areas? Are there any that deserve resurrection, if you will? And this is going to be my run into a thematic aspect of, I think it's critical that we, on a proper basis, set up directed areas of study to bring a vast array of researchers together under consortia to advance the cause. And that can be done on defined areas and with defined dollar amounts in aggregate. That was highly successful during my tenure at Juvenile Diabetes Research Foundation, that led to all kinds of breakthroughs. And the fact that people were able to work on a collaborative basis across a lot of different institutions to piggyback off of research was incredibly powerful. But one of the biggest elements was a centralized data stream of information that they otherwise wouldn't have had access to. So anyway, that's all kind of blended together, a bunch of mishmash comments there. So my apologies.
Thank you, Mark. Actually, that was helpful. Pat.
A few comments. The comments about the size of the grants was made by one of the presenters related to the discovery grants. Because the presenters were not really involved in the trans or clin areas. It's great to hear that CERM is that the team is considering and hearing loud and clear what's going on in terms of research costs, which is way ahead of the NIH, which is still using caps defined back in 1995 for the size of basic research grants. So I think it's important to consider that, because the expenses, particularly it's bad enough on the supply side, but the personnel side, there have been major changes in a very short period of time in the state of California, where nearly all of the grants go. So what used to cost, there's at least a 25, 30, sometimes 40% increase in salary. So that's really important. The other is that in the neuro area, given what we heard in the presentations and the kind of models, to be honest, I can't remember what the average length is for an awarded DISC grant. Is it 3 years? Is it 5 years? Is it 4 years? Does it vary?
Years for train.
Disc.
Disc?
Yeah,
Great.
he asked about this.
For this is 2 years, for this 0 and 3 years for these two.
Two.
And so I would say that's a real problem, right? Because 3 years, when you're growing organoids, and it takes 6 to 9 months to get your first ones, right? They don't take 2 weeks, or you don't have, you're not doing mouse work. A 3-year project is really challenging to pull this off. So we have to think about that in terms of whether a certain, and that can be done through RFAs, certain areas of emphasis, human disease models in psychiatry, for example. You can see the number of grants are like minuscule. You just can't do it in 3 years. So some of it is the dollar amount, but some of it also is the length of time, and whether the brain trust will feel that distinguishing between length of time is a reasonable process, but I don't know who can do any of these projects that we heard about in 2 or 3 years. I'll stop there.
So for these two, for example, for the development candidate, we have 3 years, but they have to come with the preliminary data. I just wanted to clarify that. So there's enough. Now for discovery, I could say, we are, as I was mentioning, going to be presenting a new concept for the neuro discovery strategy that is going to take into account the time needed to innovate risky projects and the time needed to collaborate. And we are going to be bringing this up to the board. So we are listening, we are taking into account all these different factors, and we are also looking at what other successful organizations have been doing, not just the NIH, but others like HHMI, etc. So we will be taking that into account.
Yeah, I think that's great, and I think that will stimulate a lot more activity if some modifications are made. And to be honest, pre-pandemic, you were likely more ready to run on a project once it got funded because you had folks in the research group that were already trained. Filling staff positions is far more difficult now. Training takes more time. These are all the things that are happening in the first year. You can't start that until you get the grant. So I'm just saying that it's more than just the cost of the salary costs or the resources, materials and supplies. It's also these other logistics that are extremely difficult now. So whatever you can do to adapt in a way that's really proactive, I think that's great. It'll take NIH a lot longer to do it. And some of us have had conversations with institute directors about this, and they seem to recognize the problem, but they don't really seem to be moving at more than a snail's pace.
So a sensible conclusion from this part of the conversation is that Neuro should try to lead the way on making sure that awards are big enough and long enough, and then it should percolate through the rest of CIRM's areas.
Yeah, I think that's a really good summary. And again, it can be done through RFAs, where in case there are issues about, well, why is that group getting it or whatever. But when you have specific RFAs, you're focusing, and all institutions do this, nonprofits, HHMI does it now when they used to not do it. NIH, of course, has been doing it for decades, where RFAs are used to really pull in emphasis areas, and the dollar amounts and the length of time and all those things are RFA specific. So that's something we can think about.
Good points. Judy.
Very quickly, I agree with what's been said. And the third component, in addition to dollar amounts and length of time, is % effort committed. And this came up in the same conversation that we're referencing and has come up in the past as well, to not preclude people from bringing funds in from other sources to fully fund their research operation.
Great point. So other questions about the data as presented or comments, please?
I had an answer actually to an earlier question, Larry. Do you want me to answer? I think it was Dr. Fisher-Colbury. He was asking about the research that we can see here from this idea to discovery candidate that we have not seen moving forward to TRAN and CLEAN. And the main answer here is that many of them are either still active awards, or in one case, let me see. Yes, it was mostly that they were active awards still. So we are, and in one case actually, somebody is currently applying through a company. So that could be, and our team is now discussing details with them. So this is still all alive.
Terrific.
Marv, you're up.
I was just wondering if we're doing any current outreach to institutions in the neuropsych area to see more what it might take? Because in this last discovery in the 85 new things I saw, I didn't see any that were specifically in the neuropsych areas that I could distinguish anyway.
So could we go back to one of the slides that showed funding by disease burden? Because this gets us into what I think is another important question for us to be discussing, which is it important for us to allocate by disease burden or by disease frequency or by some other metric of disease in the population? Or should we continue to be completely opportunity oriented by quality following review? And yes, Marv, your observation was correct. The burden of neuropsychiatric disease is pretty impressive. And the amount of CIRM funding that we've put in there is unfortunately also impressive.
Larry, you heard from the experts. I mean, and you know quite well that, there's a reason why industry has pulled back quite a bit, particularly in neuropsychiatric disorders. It's tough. It's tough sledding. It really is. So we have to have a dose of reality. Like the limitation on grants, the limits on grants is in large part due to the topic area itself, right? It looks better if autism spectrum disorder is a DSM-5 diagnosed disorder, so that could go into neuropsych, so it's making the numbers look a little better, but it's just really difficult. And I think we can do outreach and all that, and we can improve things, certainly, because we heard some great people who are in California, but it's really going to be slow. It's not for lack of trying, but in these areas, it's just really, really difficult. There's not enough. And I think several of the investigators talked about this who presented to us. There's still a ton of discovery to do in this area of psychiatric disorders and brain dysfunction. Just is.
Great points. Vito.
Hi, everyone. Rosa, thank you for these slides. They're very powerful, showing the discrepancy between the dollars spent, the granting, and the disease burden. And picking up on something both Marv and Mark just alluded to, if I could tease it out as a question, and Pat was just speaking to it, do we have a feeling, a quantification of how many, going all the way back to 2007, how many applications have been made to CIRM in these areas, but perhaps were rejected? That would take somebody, and if we don't have a quantity, do we have somebody with a longer institutional memory than mine that just has an anecdotal feeling like, we've seen a few of these before and we rejected them? Because I'd like to know why. Maybe it was just premature. The science hadn't developed.
Great.
I don't know whether that question goes to Jen or to Gil or Abba. I don't know. Or Lawrence.
I have a science, potentially a science explanation. When it comes to the projects with small molecules and large molecules, we always ask for the compound to be involved with stem cells or a progenitor stem cell. And for that reason, we have not allowed any small molecules or large molecules that don't have that association. And until recently, gene therapy has become more popular. But for that reason, Tran and Clin have not had any projects that dealt with neuropsych because the small molecules will have to be acting or involving stem cells. And it's very hard in the neurogenesis area. There is still this kind of lack of agreement with many of the scientists as to whether compounds that affect neurogenesis actually involve a stem cell or a progenitor. So that's just one scientific explanation as to why we don't have any in Tran and Clin.
Well, I won't
And
ask.
from this, I was going to compliment my colleague, no, go ahead, Dr. Imbashani.
No, I think you want to follow up on that. I was going to ask a follow-up question. Go ahead.
I was going to compliment the excellent answer from Dr. Creasy. In terms of discovery, we have not seen many, and the reason is a different reason. It could be the study of disease mechanisms. That was not one of our priorities, but also at the time, the science, the models were not as ready as we have now, which is some of the focus that Dr. Goldstein has had in the previous task forces to show the readiness of the field in terms of discovery foundational research. So that was the reason, and we saw a few. And in fact, one of the people that had a model that had applied was one of the people, Dr. Brennan, who used to be at Dr. Rustigage's lab. So that's one of the single people that we had in neuropsychiatric diseases applied.
But, you know, I'll point out, Vito's question is still pretty valid. Do we know that we didn't get a lot of neuropsychiatric applications in the past and then turn them down?
Yes, I asked the question. I have the team at the ready, and I asked, and we have Dr. Kelly Shepard, who has a historical memory, who's been with us for 13 or 14 years, and she confirmed that we have not gotten many applications over the years in the neuropsychiatric discovery field. Thank you, Kelly.
So if I could just close out my set of questions, I'm not going to ask this question, but our conversation is begging the question. As we expanded once before this, the stem cell definition of SERM's basis to include genetic therapy, is there room for a conversation going forward to expand it or alter it yet again if we identify such a huge need? I'm not asking the question, just planting the seed.
Thank you, Vito. A number of us have been having conversations about exactly this issue. Although I will point out that if you were to do drug discovery in the neuropsychiatric area, you could be using stem cell derived models for your optimization all the way through up until you're in clinical trials. Steve.
Yeah, so let me just follow on Vito's thought process for a moment. So as you know, Dr. Imbasiani, we did this once before, and we expanded the area in which we're involved to include gene therapy. It's not part of the stem cell area. But we did it because there's wording in Prop 71 and now in Prop 14 called other vital research. And it's a hook to hang things on that are not stem cell based. So it's, it's providing $5.5 billion in bond funding to allow CIRM to continue funding stem cell and other vital research to develop treatments and cures, And when you look specifically at the 1.5 million, there's no reference in the 1.5 million that it's, stem cell based or gene therapy based. It's just a pot of money aimed at a particular therapeutic area or a particular, organ of the human body, the central nervous system and the brain. So I do think that there probably is, although I would defer to Scott Tosher and Raphael, our general counsel, as to whether we could use this language to basically broaden the net that would bring in small molecules and large molecules, to be part of dealing with CNS disorders if we wanted to. But I do agree. I think the real issue is we're still way back at the discovery stage of trying to understand how these things work. This is in neuropsychiatry now. Sure. So, but anyway, there is potentially a pathway to broaden the net a bit if we want to. At least that's my impression.
Yeah, Steve, for what it's worth, this is Scott back at CIRM. I think your reading is correct, and the vital research opportunity language in both propositions does allow us to expand the sort of technology basis for these awards.
Thank you, Scott.
Yeah, and I agree, Steve. Raphael speaking.
Thank you.
Well, and since we are getting progressively more committed to the development of neuropsychiatric programs to try to remedy this void that we have, this may be a good area to test some of those ideas out in and see how they go. Let me just ask, is there anything else in the neuro area that has been neglected to the extent that neuropsychiatric disorders has been? Is there something else that we're missing?
Is neuromuscular included in some of the other categories?
Well, ALS and motor neuron disease is there, Vito. So that would be one, major form of muscle disorders, but that would not include, for example, Duchenne muscular dystrophy. That would come up in a different set of programs. Judy.
In speaking to some of my colleagues in psychiatry, because I am not a neuroscientist, they did ask if teams were being put together, for example, would it be possible to include experts on social determinants of disease and in specific in mental illness? And so I said I would ask the question here. Probably Marv would know more about it than I do.
I think always the initial answer is, can you get it past the reviewers? But there could be carve-outs in RFAs, for that language, if that seemed appropriate. It's something I hadn't considered, Judy, but it's a great point.
I think it's hard to imagine doing it in the model systems that CIRM is funding. And the question is whether, most of the discovery areas are using animal models. And so, and I work in this area, I think it's a major stretch. And very complicated because the genome is finite and the social determinants are not. So it turns out to be really complicated. I was going to say, given the burden and also given the prevalence, I'm surprised about, and given the possibility of using therapeutic approaches that is the bread and butter for CIRM and the initial focus, I'm somewhat surprised about the number of the amount of funding for stroke. And I don't know why that is. I don't know how the dollars are distributed in terms of the various programs, but well, this isn't DISC. It may be because the model systems are animal models, et cetera. But I think there's so much discovery on the molecular basis of cell survival and therapeutics in that area that this is another area I think would be ripe for trying to emphasize, particularly when you look at the burden. It's basically tied for number 2. And the number of grants that are listed are pretty low.
Marianne or Rosa or somebody, can we see the slides for Tran and Clin to get a sense of whether stroke continues to have that level of relative funding?
If you look at slide 13, the only stroke showing under indication is the one with Steinberg at Stanford. Otherwise, it doesn't show up as a separate category. I imagine all the funding, because if you follow that line through, we're already at Clin 2, but probably all the funding that's attributed to stroke has gone to Dr. Steinberg at Stanford. That would be my intuition anyway.
So the one that we see here, like usually stroke have larger grants. That's why we see more in Tran and Clean when Marianne showed it, and they are much more in later stage. As you can see here, we have, and I would let Abla speak, Abla opine here, but stroke in TRAN and clean is at a much higher rating in terms of spending.
Well, that's good news.
Mark.
Yeah, I want to, the question about what are we missing, I want to ask a corollary question, which I think has to do with not the disease indication, but rather the underlying root cause, because we've found out so much more in the last even 4 or 5 years around the brain-gut axis related to the microbiome, for example. It's off the charts. And the question might be, is there sufficient funding going in the study of that area? The other corollary area has to do, and again, some of that's tied to the brain-gut axis with the microbiome, but some is completely separate, is much greater involvement of the immune system across a wide range of neuro diseases than what had ever been anticipated. And I also point to the relatively recent discovery of the lymphatic system in the brain, which is a shocking thing to find out just a few years ago that it even existed. So I wanted to ask the question along those lines, as well as the disease indication lines, because some of this also gets back to consortia and other kinds of conversations that I think we need to have. So I don't know if anyone can comment on those two areas of, are they opportunities for focus or not?
There's been quite a lot of work on microglial roles in different neurodegenerative disorders, and that's a part of the brain's immune system. It's not the B and T cell systems, but it's a really important determinant of how these diseases go. And some really incredible modeling studies have appeared in the past 6 months that use organoids and microglia transplanted, for example, into mouse brains and tracking their behavior, following their in vitro behavior.
And Dr. Goldstein and Dr. Bishop-Colbury, so for the, again, the concept that we are going to be bringing in September, we are taking all of these aspects into account. So neuroimmune axis is involved in neurodegeneration, neuropsychiatric, neurodevelopmental. So we could be prioritizing, or the scope of some research, the fact that it will be multidisciplinary type of approach could be enticing immunologists to work with neuroscientists to try to quiz these kind of mechanisms and sort them out through the new program that we will be bringing up. So I hope that answers your question, but we're taking into account. Yeah, because one of the ways that we were thinking about it, go ahead.
No, please continue.
No, another way that we had thought about it, there is the microbiome, the gut microbiome, there is the vasculature, there is the neuroimmune system, but another way to look at it, so we could have looked at things like that in terms of systems involved and prioritized, but then we could be taking one over another. So perhaps the way to do it is say neuropsychiatric, and then say, take a systems approach and figure out how these systems are involved. And then we can collaborate also with people that are bringing consortia from neurodegeneration that have the neuroimmune component. So those are things that we are discussing. We are talking to different potential consortia that to collaborate in the future, and that will be brought up in the very exciting concept that we will be presenting in September.
Great, and just thank you very much for promoting me to doctor. That's quite an honor, but unfortunately I haven't quite gotten there yet. So it's something I can look forward to in the future, perhaps. So thank you.
You get an honorary doctorate, Mark.
Honorary, thanks.
I want to make a comment about the social determinants of disease and remind people that it's not necessarily the case that that's off limits to stem cell-based modeling, because it's often the case that different genomic and genetic variants make people susceptible to different social structures. A really good example is opioid use disorder, where it's very clear that there's a genetic component to people who succumb to opioid use disorder and those who do not. Did I just drop a sledgehammer on the conversation? Sorry about that.
No, well said.
We may be running down here. Any other questions about how we're prioritizing? I will point out to people also to remember that sometimes a therapy that is developed for a very severe rare disease, once it's validated by clinical trials and shown to be safe and effective, can be spread into much more common disease. So we do want to take the distributions with a bit of a grain of salt. You know, Parkinson's and Alzheimer's therapies may work for each other, et cetera. But I have to say, personally, I still find the neuropsych numbers pretty compelling.
Yeah.
So let's then turn the conversation to the other sort of issue about prioritizing in thinking about a set aside or a minimum amount that should go into the neuro area. To what degree do we want to let the current system more or less continue, which is, you know, general RFAs and then priority decisions made on the basis of review versus how much top-down intervention do we want to make in the specificity of RFAs? For example, an RFA specific for neuropsychiatric disease or Alzheimer's disease or something else because we felt there was the need.
I mean, my own personal feeling, Larry, is that RFAs can work quite well. I think the CERM team needs to think about whether they want to promote a team science approach. So, the Conti centers at NIMH, when you look at the dollar spent and the return on investment, some of the newer therapeutics came out of Conti centers. These are, 4 or 5 projects within the context of a center, and the centers can be distributed geographically. But the ones that were at Yale and Rockefeller and in Pittsburgh, they've made a lot of progress because you just make a lot more progress on very difficult problems when you use this kind of approach. So I would really favor them looking closely, and you can do an evaluation of how those Conti centers. It was named after a House of Representative person from Connecticut, but they're essentially program projects, right? And they can be clinically focused. They can be a lot of them are translational focused, so they would fit into the trans domain. But I think RFAs like that would attract, and with enough money, they would attract really high quality projects. And there's no better place than California to do that, and because of the research talent that's here.
Yeah. It's a great point. We really ought to look at other granting agencies and ask what sorts of mechanisms have they found to be very successful that we might want to import into our own processes. Steve.
And so to sort of follow up on that, and this is very much an off the wall thought, but one idea is to award a substantial amount of money. And let me just, for the sake of this discussion, to try and make it a little more tangible, is to say we're willing to provide a $20 million grant to whether it's one organization or several organizations that get together to do some research and this is in the neuropsychiatry area, to do some research that's a little more directed. So they would come up with the idea of what the directed research would be about, who the participants are going to be, and what the outcome is. But we'd be willing to provide, and this is something we don't do now, obviously, but a significantly large amount of money that's going to take more than 3 years to get done, likely, but it will make a significant advancement in our understanding of, first of all, the genetics of neuropsychiatric diseases, as well as potential points of intervention in the pathways that are involved. So again, just maybe a harebrained idea, but I think, given where we're at in this situation, it may be that we just need to really up the ante here to attract people and move the ball forward.
It's a great point. As somebody who ran a lab not too long ago, costs can just kill you, and grant rate increases year over year just don't keep pace with inflation. Marv.
I just wanted to say, I thought Stephen's idea was great. I think we should consider something like that.
I can answer that. So for neuropsychiatric discovery, we are developing the concept that we've developed that we are going to be bringing is responding to this idea. So you will see it structured in that way. And I think it's something to be very excited for. The second thing is in terms of program project grants, which are multidisciplinary, long-term, we have taken the experience that we have as a team. We've all worked in different, so I personally work with the NIA and the NIMH and the NINDS in the different accelerating medicines partnerships for schizophrenia, Parkinson's, and Alzheimer's, which were consistent of program project grants. And we've taken that experience and we've actually implemented it in the structure that we are going to bring to in September. So again, I think we hear that loud and clear for Discovery in Neuropsych.
Mark.
You say Mark or Marv?
Mark.
Yeah, I think those are both fantastic comments from both Stephen and Rosa, and I think that's exactly the kind of approaches that we need to have a concentrated game plan and to have multidisciplinary groups involved, because it's gonna require different ways of doing things than just random people submitting proposals in for random study activities. So I concur on both of those comments, and both well said. So thank you.
Great point. Other thoughts about these issues of specifying priorities versus passive review? Well, passive, semi-passive. So Mark, go ahead.
Yeah, we haven't really addressed the first question, right? Is that something we want to try to take a stab at, or how do you want to frame the conversation around number one there?
Well, let me just throw out a possibility. I mean, it seems to me that by and large, the broad RFA review-based system for setting priorities does a generally good job. It doesn't hit everything by disease burden necessarily, but the flaws, if any, is that it sounds like grant sizes just haven't been big enough, and grant terms need to be a little bit longer to cope with some of the experimental challenges in some of these areas. My personal view would be that we should do what we're doing with neuropsychiatric, which is we've identified an area that is obviously woefully under involved with our agency and needs work, and continue to be alert to things that we're not doing that look like they should be profitable, but that otherwise I would not be in favor of substantial programming of the entire $1.5 billion, say. I think we have to rely on the community to tell us what's scientifically exciting, valid, and feasible.
Is that something, though, that maybe a third might be a consideration for something like that, that's a little bit more directed to be able to pull people together, or is there some other, number that might make sense?
Great question. Somebody else want to take a stab at this?
I mean, I personally feel, Larry, it's hard to think of a what size of the pie, what piece, without seeing the RFAs. I mean, I agree with you that it should not be the whole nut, right? It can't be. For all the reasons that you said. So I don't know where to titrate this, but I think the idea that we've heard about really putting an emphasis on pushing teams and funding at a higher level because of what we're faced with, not CERM, but families and individuals who are suffering, is the rationale for really ramping this up. But I don't know what the number is. It shouldn't be all of it from my perspective, because I agree with you that you've got to have some grassroots development. You never know where a great idea is going to come from. So we don't want to be prescriptive for it all or even a majority, but I do think it could be a reasonable size of this, and it would make a huge difference.
Larry, we have a couple of comments here from the CERM team, if that's okay.
Oh, yes, please. Oh, of course.
Thank you so much. So the team has been looking at kind of what our historic spend has been in terms of percentage of the discovery programs kind of coming in through the pillar program. And based on the estimate of what Rose and the team are working on for what a concept for this collaborative research model, like the program project grant like model, and her estimate for what that would cost all in versus what the target is for translational clinical stage programs. We think that it is very feasible to still continue the pillar programs, meaning the ones that come in organically, and be able to have enough to accommodate a good amount of those, plus probably fund, if the board approves to the scale that Rosa may propose, probably 4 big program projects, plus still be able to support between 7 and 10 translational clinical stage programs a year. And then just as just the back of the envelope, that would still be accommodated by the minimum of the $1.5 billion in spending. Of course, more of the research budget could be utilized if there was some promising direction. The board could decide that more of the research allocation can be put toward the CNS programs. But I think that everything that the board members are proposing today could be accommodated. It's they're a combination of utilizing this standard pillar programs plus a more directed significant investment in these program project like grants while not compromising the translational clinical portfolio.
Those are great points, Maria. And, we could look at it as, we're taking neuropsychiatric as an example and we're building it up. And we can see, what the rate looks like at which spending is going to need to increase to keep in step with demand and burden.
Right. So this is just charting it out based on, just forecasting, let's say, in the instance of success, and you're able to fund 4 of these big programs, you'll still be able to accommodate the organic inflow of new discovery research at pretty much the same rate if we're accounting for current allocations and still translational clinical programs. But Albla Creasy, who heads our therapeutics development program, wanted to add something to that as well.
I just wanted to go back to what Mr. Josgard mentioned, the vital research opportunity. Would the committee consider, with your leadership, Dr. Goldstein, thinking of small molecules and potentially large molecules as potential new opportunities to fund for Tran and Clin as a vital research opportunity to complement what is going to happen with discovery?
I think it's certainly something we should look at, although I do think that the use of stem cell models to be optimizing grants, optimizing progress, and to identify subpopulations that are likely to respond to a given small molecule is a way to keep it in the ranch at least a little bit. So
But as pointed out with the discussion we had in the second Neuro Task Force meeting, it's going to take several years before such molecules or such compounds are likely to score and move to TRAN. And as you know, TRAN takes 2 to 3 years, then CLIN takes 4 years. We can have both options. It's your choice, obviously.
It's certainly an important discussion for us to continue. I think the other question is going to be the use of primarily novel chemical entities, NCEs, which is, of course, the way it's done in industry because they're patentable, versus continuing to mine through existing drug-like molecules, FDA-approved drugs that have been proven to be safe but didn't work well for the chosen indication, et cetera, et cetera. David Higgins.
Yes, this is probably obvious, but do we have any way of knowing, or can we have a way to know why grants that are initially approved fail? They either don't come back, presumably because they have some insight as to their progress, or they resubmit in the next stage and they're just not a high enough level to be approved. I mean, it seems like we're willing, everything's on the table. I'm hearing all these positive reception on the staff's part to changing the way we do things. And can we change the way we do things and include why have people failed in the past?
Boy, is that a good question. And personally speaking, there's a lot of different ways to fail.
Great point, Larry.
I don't know. Has the CERM team done any analysis of this retrospectively? I'll make the question.
We don't have, Larry, can you hear me? The
Yeah,
team
go ahead,
is,
Maria.
yeah, so the team has been, we certainly have, based on what you heard from Rosa's report, not enough, in terms of projects in the pipeline, and some of them are still ongoing. But as you know, for traditional drug discovery, when you have, even for, small molecules, you start off with 10,000 compounds, and at the end, you'll have one product, right? So it's just part of it is the nature of what, the path is from discovery all the way to translation into clinical. I mean, I think that the kind of the value proposition or the prospect that we have that's very exciting is that when we have a therapeutics that's developed in the cell and gene therapy portfolio, it's a definitive treatment. So that is, the impact factor and the value of that is wonderful once you have that. So I don't think it's unusual to have many programs in the early stages where it really is just looking at the science as in this is scientific research. Not everything works out. It may lead down a different path. Same thing with mRNA, how mRNA was first looked at for a variety of indications, including being able to have a better way to reprogram iPSC, right? And then now, it's utilized for vaccines. And now we saw an early outcome from an early Parkinson's, pancreatic cancer trial with mRNA. So I think it's a matter of it's really difficult to view it project by project. I think if you are able to tag these things and then follow them through its course, one may see that it would lead to a different type of therapeutic. So it's not, something that is really easy to analyze, but I think that's the reason why basic and foundational and mechanistic research is very important, because you don't have an answer yet, and it gives rise to potential, to knowledge to lead to answers. But where we can track a little bit better is when it's in the kind of the later stages, translational to preclinical to clinical. There we have a better accounting for why things fail. So we have a really good sense of kind of some of the hurdles, and those we're trying to address through some of our programs and how the teams are managing them through manufacturing initiatives, through, regulatory type approaches. And so those are the types of things that Abla has presented in kind of her presentation. It's still not 100% obviously that we can solve all the problems from translational to clinical, but we have a better idea in the later stages.
But we have not had neuropsych therapies in our TRAN and CLIN programs, just to be clear. We have not had any apply that failed or succeeded.
Of 0%, we have a 100% success rate of them not progressing.
Not progressing because they haven't applied.
Because we don't have them. And I think as a general question to Dr. Higgins, to your general question, it's just the nature of kind of discovery research to translation to clinical. We do not expect that the progression should be very high from the discovery into translational. But some of it is actually not intended to give rise to the product per se. It may start you along a path where it leads to downstream a therapeutic candidate, even if not directly.
Yeah, when you talk to people in the trenches, they'll tell you it's harder than it looks. Other comments or questions, or should I summarize and wrap us up?
What's the timeline for the team presenting? Rosa, you mentioned multiple times we were going to see a variety of different proposals.
Yes.
I don't have my schedule in front of me. So what's the timeline?
September.
Dr. Levitt, I think Marianne and Scott have so I believe the first presentation to the science subcommittee task force is either is in early September or in late August. Is that correct, Marianne? Late August? I know you are scheduling it still.
Yeah, I think that's the general timeline, but there hasn't been anything confirmed yet.
Okay, so you're looking at the last week. You're looking at the last week of August, right?
The last 2 weeks of August and the first 2 weeks of September were the first week of September, I believe. That's what Marianne and Scott were looking into.
All right, thank you.
That could be the concept to the task science subcommittee before we come to the board at the end of September, just to clarify.
Yeah, that makes sense.
Yeah, great. Thank you.
Exciting.
Yes, it is very exciting. And it sounds like you've done an enormous heavy lift. This is really a tough area. So even before I see it, I'll send my congratulations to the team. This is really
the team. Takes a big village, and thanks to all our colleagues. Yeah, thank you.
Yeah, no, you guys have been doing a great job. So if I were to provide tentative answers to these questions from the task force, what percentage of $1.5 billion do we want to make specific recommendations for? Sounds like the answer is not necessarily a large fraction, and that the thing to do, at least to start, is to get into an area that we haven't done before, make sure the awards are substantial, and see where it takes us. And as to other important areas of neuroresearch that we're missing other than neuropsychiatric, Pat made a good comment about stroke. It's something we ought to have another look at some point. We can have experts come in and talk to us about what's going on in stroke. I think that's something that we could do. But certainly looking at the neurodegeneratives, it seems like we have pretty substantial investments, and that the amount of them is reasonably related to what I at least know of opportunity. So do we have public comment as part of this?
Marianne?
There currently aren't any right now, Larry.
Good. So unless there's an objection, I'm going to move that we adjourn. I'm going to point out that we should let other members of the task force know that this recording will be up on YouTube. They should look at it and let us know what their ideas are. So otherwise, thank you all for your time today. Valuable conversation, and we will see you next month.
Thanks, Larry.
Bye-bye.
Bye-bye.
Bye, everybody.
Great work,
Thank
Larry.
you.
Thank you.
Great work, Larry.
Right.
Mhm.